Hi everyone, good morning. I'm Tahmini Hossain and I'm a PhD student in South Dakota State University. So I basically work with antibiotic tolerance, persistence, and resistance. So before talking about minimal cell, I'd like to give you guys a brief idea about the background of what I am doing and why we are using minimal cell. So I think all of you already heard about antibiotic resistance. It's one of the current global threat. So although we know it's a very serious problem, but we don't often realize how serious it is. So that's why here I am showing you guys some statistics. So in each year, 2.8 million people contracted an infection that does not respond to antibiotic, and over 35,000 of them die. And in Europe, 33,000 people die each year, and this is not only prevalent in US or Europe, this is prevalent all over the world. And recently I saw a very recent report like where they have estimated that by 2050 each year 100 million people will die because of the antibiotic resistance bug. So now I think you guys realize how serious this problem and uh, how uh, how it's important to get rid of this problem as soon as possible. So although we talk a lot about antibiotic resistance, but we often ignore like one of the common term which we call antibiotic persistence or antibiotic tolerance. So what I'm trying to say is that antibiotic resistance is not the only way to survive during antibiotic treatment. Bacteria can survive antibiotic treatment, just no genetic changes, just doing some phenotypic changes. For example, like Superman, like without his dress he's just a normal human being, but with his fancy dress, like he has some superpower and he can defeat the bad people. So bacteria can also do the same kind of thing. They can go through a phenotypic change or metabolically repressed state to just escape antibiotic treatment. So let me just go through with you this figure. Uh, I don't know like where is the option for that. Okay, let me just go through with this figure. So if you can initially see like when we treat a cell with antibiotic, so okay, so when you treat a cell with antibiotic, like only a few percentage of population can survive the antibiotic treatment, um, just going through a metabolically repressed state. And when the antibiotic treatment period is over, this population, the blue color cell, which we call the persister cell, they can revive back and they can again uh, they can again make the whole population again. And this is one of the main reason behind the reoccurrent infection. But the main critical point is that when this population revive back, there is a high chance that this population can gain resistance, or they can mutate and gain resistance. Like if you can see the red cells, and once they gain resistance, like there is no way the antibiotic will able to kill them. So so our lab is trying to figure out, like in my project, like I'm trying to figure out like how this persister cell form, or how bacteria can sense the stress and they go into that metabolically repressed state, and how they maintain that repressed state over the antibiotic treatment period. So we initially started working with E. coli. So what we have done, like we isolated the RNA from the general population and we isolated the RNA from persister population, and we have observed like what genes are upregulated during the persister cells. So we did a complex transcriptional data analysis, regulatory network analysis, and I'm not going through all the results. Like I'm just giving you guys a very brief idea like what we have found. So we have found that antibiotic response is not occurred by a single mechanism. There are multiple mechanisms exist, and this is not, we have found like uh, this antibiotic persistence is studied in the last 80 years, and there are several research groups, and different research groups say like different mechanism. For example, ATP synthesis or ribosome hibernation genes are the essential for antibiotic persistence. So from our initial result, we could not able to figure out like which are the essential mechanism behind antibiotic persistence. So we thought like if we can study this antibiotic persistence using the minimal cell, it will be very intriguing, or for us to sort out the mechanism, because Syn3B has very less number of genes. It has only 473 genes in comparison to E. coli, and also it contains mostly essential genes and networks. Another important factor is that the Syn3B lacks several genes which previously people have found related to persistence, for example, T systems or ribosome hibernation genes. So as you already know that working with Syn3B, like one of the most difficulty is that like Syn3B does not have higher growth rate. So I initially started to work with how I can increase the growth rate. So I initially optimized the media, and then after optimizing the media a little bit, although it did not help that much, so I started to evolve the population. So how I did that, I just grow the Syn3B until exponential phase, and after that, from the exponential phase, I again dilute the culture and again grow them to the exponential phase. So in that way, we go around 26 cycle. So what we have observed that this evolved cell, which we named as PO26, it has higher growth rate in comparison to the Syn3B. So we basically um, uh, observe the growth rate based on the optical density and based on the CFU per ml. So what we have found in Syn3B, the growth rate, the doubling time is around 6 hours, but for the PO26, the doubling time is around 2.5 hours. Then we ask ourselves like whether this minimal cell have antibiotic tolerance and persistence. So before showing you guys the result, I would just like to talk about how we define antibiotic tolerance and persistence. We define antibiotic tolerance and persistence by using a biphasic death curve. Like if you see the death curve, when we give population antibiotic, like the cell will start to die. So for example, if you see the green mark, like so the initial population or the tolerant population, they have the death rate higher than the second phase, like the red mark line. So in the second phase, the cells started to die slowly. So we call the second phase is the persistent phase. So we wanted to see whether minimal cell also show this kind of graph or not. And what we have found that yes, minimal cell also show the same biphasic death curve during the antibiotic treatment. Like if you see in both from the exponential phase and from the stationary phase, like around 24 hour, the first initial death rate is higher in comparison to the second death rate, like 24 to 72 hour. So from this result, we can see and we can say that minimal cell have both antibiotic tolerance and antibiotic persistence. Then we wanted to see like whether how fast minimal cell evolve, as it has a very small number of genes. So we thought like minimal cell will evolve faster than other organisms such as E. coli. So this project like done by one of the undergrad from our lab, like Eliza. So what she had done, so she grew the culture Syn3B until stationary phase, and then she diluted it into the antibiotic containing media, and then she treated the cells with 24 hours with the antibiotic, and then she washed the culture and do the same cycle again, and she observed how many cycles it basically take to for Syn3B to gain resistance. And what she has found, like it takes very small number of cycle. For example, for streptomycin antibiotic, it takes only 4 cycle, which for E. coli it takes more than 20 cycle to gain resistance. And not only one antibiotic, she also tried 2 antibiotics, streptomycin and ciprofloxacin. It takes only 9 cycle, and she also tried with 4 antibiotic, which just takes 16 cycle to gain resistance. So from this result, we can say like minimal cell can quickly evolve resistance, as it has it has a very minimal number of genes. That's why. So now we are working on to evolving minimal cell under extreme stress condition, for example, high or low temperature, which we already have done. One of our undergrad from our lab already done. And what she evolved Syn3B, like she evolved Syn3B at high temperature 42 degrees Celsius and at low temperature 30 degrees Celsius. And we are also trying to evolve them like under high or low pH or high or low salt concentration. And we will start a very new project, like we are thinking, so where we will evolve minimal cell to grow at low or zero gravity. So that's all. Lastly, I'd like to acknowledge my lab mates and especially Eliza, who did all the evolution experiments. Thank you so much, and I am open for questions.
I mean, Tahmina, what was
Yes.
the main difference between SP4 and SP16?
So, like for SP4 and XP16, like some of the component we basically increase the concentration. Um, for example, what we called it 2XP1. So the XP1 we basically increase the twice concentration, like tryptone, yeast extract. We increase the concentration, and we also added vitamin B1. That's what we have changed. Yeah. So I can send you the link of my paper. So in there,
I was just hoping that you would explain this for people who haven't read your paper. I have. This was.
oh yeah. So yeah, we increase some of the components. I mean, we increase the amount, and for some of the and we added vitamin B1, which really helped to for the growth. Like that's what we observed.
And then have you worked out to send your send cells to the space station?
What? No,
I don't know how else you would do zero gravity.
Oh no. Like that's what we had. We did not yet started that project. Like that's what Dr. Nicholas is trying to collaborate with School of Mines, and they have a machine. So I mean, in that instrument, you can actually grow the cells like at zero gravity. So they work with NASA and they work with the space microbes. So we thought like if we can evolve like minimal cell in zero gravity, like that will be like very cool. So we did not yet do that, and I did not yet see that instrument. Like we are planning to do that.
Then last question. So
Mhm.
you said that Syn3B has a doubling time in your
Mhm.
media of 6.5 hours. So we
Yes, around
find
six
that
hours.
it grows that it divides in about 2 hours. Are you
Mhm.
exposing it to tetracycline? I'm
Yes.
okay. That makes perfect sense to me. Why are you using tetracycline since you're not? I mean, why did you continue to use tetracycline?
So we are using tetracycline because like to I mean, I think like just to avoid the contamination. And
Okay.
yeah.
Okay, that makes sense.
And also like uh, the doubling time we have measured, like we used optical density and we use CFU per ml. We first tried to use the DNA dye, but it did not work very well for us. So that's why we use that method.
There is a video made by Kim Wise on the Minimal Cell website if you want to see how he does his doubling time measurements. That is very clear and I think might enable you to do that his way if you're interested.
Yeah.
Okay, thank you very much.